morning, good morning, good morning. Hey, we're going to go through a PowerPoint today. We're not going to go through every single slide. Hopefully, just go over some points that need a little more discussion. Let's see, a little history about Gregor Mendel. Things like these you can read on your own, or you should have already read. Let's get through some other points here. You'll have a copy of all this stuff, so that's good. Why use the peas? They were a great subject to use. They had a short lifespan, so many generations in one season. The traits were either or traits. He could control pollination. And here are some slides basically on how to control pollination. He'd snip off the stamen so he could control, using a paintbrush, which pollen went to which stigma. So let's go a little bit here. Mendel's work. He used his 7 traits or 7 characters, like plant height, and most of those would either have, or they would have just two traits. They would have like a tall or a short, purple flower color, white flower color. So he's very lucky, or maybe you throw out the bad data, some people think, if something didn't agree. We'll talk more about that later. Monohybrid crosses, or the crosses that just one trait at a time. A tall plant with a short plant. Or you could cross purple stem versus white stem. The P generation stands for parental. That's your first one. We're going to see P generation, F1 generation, F2 generation. So the P stands for the parent, or the original generation. F1 is the first filial. If any French people out there remember the word for son, fils, F-I-L-S. The F2 is the second filial generation, which basically would be your grandkids. You have parents, children, and grandchildren. Those are your 3 generations that for the most part we'll be talking about. As you can see, so I got a pointer here somewhere. As you can see right here, I'll use my pointer a little bit later. Didn't matter male or female, the plants, because it didn't exist. So we'd have the generations. We'd have the first, you cross purple flower and the white flowers. First generation, only purple. What happened to the white? Did they disappear? Well, they would reappear if you get rid of my little thing here. Can't give her my video screen, but they did reappear down here with a 3-to-1 ratio, consistently 3-to-1 ratio. So Mendel's going, there's something going on. Here are the different characters that Mendel studied. You can take a peek at those and notice the ratios over here on the side. They're almost all 3-to-1. Not exactly, but very close. There is some random variation. So Mendel would think that there's alternate versions of the genes. He didn't call them alleles, we call them alleles now. And he would have one from each parent, one allele from each parent. So you'd end up with 2. There's the dominant alleles and the recessive. You should be familiar with those terms. So Mendel had his law of segregation, which basically that's a huge law. Essentially, every organism has 2 characteristics for a trait. Big P in this case. Law of segregation means that one of these P's is going to be passed on to the next generation. And same way for those flower. There's 2, but they're going to get segregated. They're going to get separated. So only one will be passed on to the next generation. So the next generation also has 2, kind of like our diploid situation going on. So that was Mendel's law of segregation. You can follow that up more in the book as well. Then we had some terms, phenotype and genotype, you should be familiar with. Phenotype, physical appearance. Genotype, what the genes are. Other big vocab terms, which you should have already completed on your assignment. Homozygous, 2 alleles are the same. Big T, little t, doesn't matter what they are. They could be small q, as long as they're the same. And then heterozygous, 2 different alleles. So the cross that he's talking about, you can have, for example, big T. Let's get the other ones up there. Can be crossed with homozygous recessive, homozygous dominant, or even heterozygous. So those are the 6 different crosses that he's talking about. A test cross. So the reason for a test cross is, let's say you have a plant that has dominant allele, a dominant trait, purple flower color. Is it heterozygous or is it homozygous? We don't know. We cannot get in there and look at the genes. However, we can perform a test cross. A test cross is you're going to cross the unknown, in this case, with a homozygous recessive. And when that happens, there's 2 possibilities. If The unknown is homozygous dominant. If you do the Punnett square, you're going to see they're all going to end up purple. If it's going to be heterozygous, you're going to end up with half of them being purple, and right here behind my lovely, lovely face is going to be 2 white. I can't even draw on my video. So underneath my picture here, as you can see on your slides, they're going to be 2 white. So it'll be half purple, half white. That is what a test cross is for. Dihybrid cross, crossing 2 traits at once. Now, obviously, you're going to have 2 different codes. In this case, tall and short for heterozygous, and this would be round seed and wrinkled seed. So basically, cross both of those at once. We'll have some more examples on the dihybrids. This just says how many possibilities are there. Let's say you're just crossing 2 traits. So it's 4 different possible kinds of gametes this can produce. Now, how do you figure out those gametes? Remember the FOIL method. Big T, big R, that's one type of gamete. Outer, big T, little r, second type of gamete. You can also have a little t, big R, inner, or you can have the last of each pair, little t, little r. So 4 different types of gametes. So if you do a trihybrid cross, you'd have 3 traits at once. Maybe let's throw in, let's say, how about yellow and green seed? You would have 2 to the cubed or 8 different gametes possible. We're not going to do trihybrid crosses. Your book talks about figuring that out a little bit. Feel free if you want to check it out. So there are 4 types of gametes. So this would be our 16 square, 4 by 4. And our ratios, not 3 to 1, but 9 to 3 to 3 to 1 would be our phenotypic ratios for this is both parents being heterozygous for both traits. 9 to 3, 3 to 1. Now the law of independent assortment, Mendel's second law. Basically it says whatever one trait is, you're no more likely to get another trait. Purple flower or white flower has no bearing on whether the plant is going to be tall or short. They are not linked. Later on, there'll be some linkages for non-Mendelian. Now for the rules of probability, this is how we figure things out. Basically, flipping a coin, get heads, 1 out of 2 chances, 50%. Rolling a dice, 1 out of 6. Why is that important? Because we're going to try and figure out what the outcomes are. If you have things, let's see, you can read these on your own. Some of these slides here we're going to really skip through. It's basically if their traits are two situations are totally independent, such as rolling a dice. If you roll a dice, what's the chance of rolling a 6? 1 out of 6. What's the chance of rolling a second dice, also rolling a 6? 1 out of 6. So you're going to multiply those together to find the overall probability. So chance of rolling 2 6s, multiply 1/6 times 1/6, you have 1/36. So 1 out of 36 chance of rolling boxcars. 2 6s. Same thing for rolling a 1 and a 7, a 1 and a 3, 1 out of 36. So this gets on the probabilities. This is kind of doing the same thing with dihybrid crosses. It's getting a little more complicated. You can read that in your book. We're going to skip all these. You do the same thing with the Punnett squares to get these solutions. So we're going to get those. Moving on. Excuse me. Law of independent assortment. So what's that saying? One trait does not depend on another. If you have blue eyes, what's your chance of having blonde hair? Doesn't have any bearing at all. It's just as if you had brown eyes, same chance of getting blonde hair. That's a law of independent assortment. Again, there will be differences in the non-Mendelian coming up, which will be the next chapter. Law of independent assortment. Here's independent assortment. This is what really happens. This is the true side. If you have yellow and round seed and cross that with a wrinkled green seed, independent assortment means we're going to have yellow and round, or you could have yellow and wrinkled. This is the false side. If you had dependent assortment, which is not true, you would have only yellow and round and only wrinkled and green. But instead we have this situation over here where it is independently assorting. So for the ratios, when we do a Punnett square, we don't predict what will happen, but what is likely to happen. Remember Mendel's slides from earlier on, or his results from earlier. He didn't get a 3-to-1 ratio. He came close. So these ratios are a prediction of what will happen. Our famous 3-to-1 ratio, 75% chance of having the dominant trait. Could you have, out of 4 offspring, all 4 have the recessive trait, like a 0-to-4 ratio? Absolutely. But over time, with enough data, which Mendel had, excuse me, it'd be more likely to come up with that 3-to-1 ratio. So dominant and recessive. Dominant doesn't destroy a trait, it just hides it. It doesn't subdue, as a slide says, it just hides it. It masks it, would be a better word to use. It still has the original trait, it still has the original allele if that allele was passed on, and it may appear later on, which is why sometimes we say traits will skip a generation. The allele was always there. Or you might have a child with red hair with nobody else had red hair. Well, 7 generations ago, that red allele could have entered the family and stayed hidden for 7 generations. And now all of a sudden, there's a red-headed child. That's the way that works. Okay, exceptions to Mendel. Mendel had everything dominant and recessive. Excuse me. Let me start a different slide on this one because it's a slightly different topic. So moving on to video number 2.